Yeah, it's like a rich.
Gotta do technology.
How are you today?
Good, how are you?
Doing well. Get the technology to work despite of all. So when you see that,
We're good. All right, good evening, everyone. Hopefully that is the only time you will hear that sound this evening. Welcome to the 11th Annual Wisconsin Science Festival and the 8th Annual Big Ideas for Busy People. I'm Eric Wilcots. I'm the Dean of the College of Letters and Science, and I will be moderating tonight's Big Ideas for Busy People as we get along the way. This is, as I say, the first festival of our second decade. That's the way we like to think about it. We had this little thing last year. So, a couple of things before we get started. I'd like to give a big shout out and a big thanks to all of our sponsors who have generously given to make the Wisconsin Science Festival possible. The other thing I'd like to do before we get started is to recognize that UW-Madison resides on the ancestral Ho-Chunk land, a place their nation has called Tejope since time immemorial. In an 1832 treaty, the Ho-Chunk were forced to cede this territory. Decades of ethnic cleansing followed when both the federal and state government repeatedly but unsuccessfully sought to forcibly remove the Ho-Chunk from Wisconsin. This history of colonization informs our shared future of collaboration and innovation. And today, all of us at UW-Madison respects the inherent sovereignty of the Ho-Chunk Nation, along with the 11 other First Nations of Wisconsin. All right, so what are the ground rules of Big Ideas for Busy People? Each speaker will have 5 minutes, and we are going to count down the clock. You will be able to see the clock on the big screen so you can follow along. If the clock hits 0, we will sound the gong. We will then reset the clock, and there will be 5 minutes of questions. For those of you who are watching online, please type your questions into the chat, and Laura will try to read those out. We're going to try to get through as many questions as possible during those 5 minutes of questions. So please, we've asked the speakers to keep their answers succinct, and we're going to ask you to keep your questions succinct so everybody can have a chance to answer a question or ask a question. If you wish to ask a question, please line up at the microphone here in the center of the room. Okay, so that's rule number 2. And after that, 5 minutes, we'll be on to the next speaker. The other great thing about Big Ideas for Busy People is it brings together lots of really fantastic folks who are doing lots of really fantastic work. And so to keep things lively and keep things moving, I will not be introducing the speakers in between their talks. So pay close attention now. Number one will be John Hawks from the Department of Anthropology, who will talk about, where did we come from and who are we? Second will be Amy Barger from the Department of Astronomy. How do we know how old the universe is? Third, Phil Newmark from the Morgridge Institute of Research, or for research in this very building, and the Department of Integrative Biology. How do we know how animals develop? Fourth, Hilary Dugan from integrative biology. How do we know climate change is real? Lakes as a chronicle of our changing planet. Next up will be Keith Findley from the law school. How can we really know who done it? The Wisconsin Innocence Project. And last but certainly not least, and fitting in with the theme of the overall science festival, Anne Pringle from the Department of Botany. How can we know where fungi are, and how can we know where fungi are and aren't? So those are our speakers tonight for Big Ideas for Busy People. And so without further ado, ladies and gentlemen, let's get started.
There we go. The story of human origins is one that usually people tell from some beginning up until now. And this is the same problem as you see with American history in schools, right? Is you teach the first colonists, you teach the Revolution, you teach the Civil War, you teach World War I. Now World War II, there's not enough time. Oh my gosh, nothing's left, right? And that's the way that it works with human evolution. It's a long history. Our common ancestors with other primates, our close relatives, the chimpanzees and bonobos, lived about 7 million years ago. And to account for all that history, you run through it and things change. Well, I've been doing lately something a little bit reversed. I've been starting from the present and looking back at the past. And so I've prepared some graphics that start from a center point. And in this graphic, you'll see the center point is the two chromosomes, the two copies of a chromosome that you have. You've got 23 pairs of those, and this represents one of those pairs. And on either side of it, flanking it, are the chromosomes of two biological parents. And flanking them up and down are the two chromosomes of their biological parents, biological grandparents. Now, in humans today, we reckon our genealogy in many different ways. We have this sort of biological genealogy with biological parents, grandparents, and so on. We have cultural genealogies. We have adoptive kin and figurative kin, and we reckon our genealogies in lots of ways. All of those ways are important. They all connect us to each other. And what the biological genealogy gives us is one little component of that story. But what I want to share quickly is how immense that component is. Because as we run outwards, you see the great-grandparents and the great-greats. Each concentric circle is going another generation back in time. In the parents, they're flanking that center pair of chromosomes. You see that one of those chromosomes is reddish and one is purplish. And the biological parents, you'll see that they're broken up into little chunks because recombination in your biological parents from their parents' chromosomes is what generates the chromosomes that you inherit. And so you get chunks of chromosomes from your parents. And they got chunks from their parents. And as we trace that outwards, these little colored blocks that I've put in here trace those chunks that are in your genome into different ancestors. Recombination breaks up chromosomes in a limited number of places each generation. And that means that as we go further and further back in time, those chunks don't break apart as fast as the number of ancestors increase. And so as we go out to the great-grandparents and the great-great-grandparents, the number of ancestors is here indicated on the left side in these blue numbers. 4 ancestors in your grandparents, 8 in your great-grandparents, 16. It doubles every generation. So by the time we get out to the great, 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 great grandparents, that's 6 generations in the past, you've got 64 lines of genealogy going to those places. But on the right side, I've indicated the number in this chromosome's genealogy of ancestors that have genetics in common with you anywhere on this chromosome. And as you see your grandparents, you've got pieces of their 4 chromosomes. And as you go further back, you've got all 8 great grandparents. But in the next generation, all 16 of them haven't given you DNA on this chromosome. Only 12 of them have. And another generation, only 3 more, even though the number of your ancestors has doubled. In fact, as we go further and further and further out, what you discover is that most of your ancestors are not genetically related to you. Now, all of us are related to each other genetically. We all are distant cousins of each other. But as we get out, this 6, 8, 10, 12 generations, the number of lines of genealogy becomes immense. We start to cover 1,000 ancestors in 10 generations, 2,000 in 20 generations, excuse me, 1,000,000 in 20 generations. In 30 generations, 1 billion lines of ancestry. And there were not 1 billion people then, 30 generations ago. That means that these lines are converging in the past. They converge into a giant network. And that giant network connects all of us. So when we ask, where did we come from? We all came from the same place. Our ancestry is totally shared. And as we look broadly across that ancestry, we'll find that the little pieces that still remain each trace a different history.
All right, thanks, John. Please, if you have a question, line up at the microphone. Laura, do we have any questions online? No questions online. Oh, we got a question. Yeah, please.
If 62,000 years ago, perhaps, we were all in a very small grouping, and then there was the great diaspora. Is present day modern humanity an artifact of a warming period in a glacial cycle of 110 to 125,000 years?
This is a great question, and I actually have a slide about this. So when we move further back into the past, what you discover is that almost all the populations in the world do converge back to an African population that lived sometime before 100,000 years ago. But that African population we've begun to appreciate over the last several years is much more diverse and is home to a greater diversity than we thought maybe 30 years ago. Every population that has indigenous ancestry outside of Africa today does stem from a very small group that lived sometime before 50,000 years ago. That small group was one of many populations in Africa at that time. And that small group spread and mixed with some other kinds of people. Those signs of mixture are actually throughout our genomes. So on the right here, I've listed, these are present day human populations that we have genetic samples of. And this is a minor representation of human populations. So 6,000 languages spoken in the world. This is just a small group of them. But I've chosen them for diversity because they have different histories. And those different histories, while they're all intertwined, go back to this out of Africa event. When we look inside of Africa, there's actually deep roots. And those deep roots go back to populations that we have yet to discover. Today with genetics, we can trace into ancestral populations and know that here's the connections. They go back 300, 400,000 years, mixing with other kinds of populations. It is a giant network. And it is that network that connects all of us. So it's a really, it's an exciting time to understand that there's this complexity.
Come on up to the mic.
Sorry. This is exactly what I've been puzzling about lately. But so there's you, and then your family tree goes out. And at some point, certain people go back to King Alfred. So at what point does your family tree start to move back in? And also, if I've got 64 ancestors, why do I only have DNA from like 3 of them?
The answer to that one is quite simple. It's because the chunks are breaking up more slowly than the number of ancestors is increasing. So many of the lines of the genealogy are your genuine ancestors, right? This is a logical phenomenon. Every one of your ancestors has two parents. That increases over around 8 or 9 generations in today's American society. If you look back in the past, many people start to trace that to fewer ancestors. So inbreeding, your parents are related to each other. All of our parents are related to each other. But the thing is that most of them don't know how they're related, right? It's far enough back in the past. Out of my 4 great-grandparent, right? That great-great-great-great-grandparent, I've visited the grave of one of them. I know where a couple of them are buried. The rest of them, I know something about through genealogical records, but going further than that, most of us don't. It's in that time frame, actually, that we start to see the lines converge. By 3,000 to 5,000 years ago, every human on Earth shares an ancestor. And as we go further back, the group of our ancestors converge into the same group. And at that stage, we all have the same ancestors, but we may inherit different genetics from different ones of them because of the lines passing back through different populations.
Real quick question. You got 40 seconds for question and answer.
I can answer this one.
Could you give us a quick update on your work at the New Horizons Cave and the Homo naledi research you were so famous for a decade ago?
That's a great question. Is the gong coming? I can give you a quick update. We've actually been very busy in the cave, and we have some really remarkable discoveries, including one that's going to be in the press in a couple of weeks. So keep your eyes out. We have a really exciting one coming.
That is anticipation. Thank you.
All right, thank you.
Hey, Amy, we'll reset the clock and we'll get you to your first slide. Go.
Oops, I think I went too far. All right, humans have a fascination with the ages of things. Let it be the Dead Sea Scrolls or movie stars. We can set a lower limit to the age of the universe from the ages of progressively older objects within it. So for example, the universe has to be as old as you, but that's not a very interesting limit. Next, we know that the universe has to be at least 230 million years old because we have dinosaur fossils of that age. Furthermore, the universe has to be at least 4.4 billion years old because we have moon rocks that the astronauts brought back that have been dated to that age. And finally, the universe has to be at least 12.8 billion years old because those are the ages of the oldest stars. A galaxy is a gravitationally bound collection of stars, gas, dust, and dark matter. Here you can see an image of our own Milky Way taken from Chile. And you may notice my laser pointer isn't working, but you may notice that there it is, that there's the large and small Magellanic Clouds visible in this image as well. However, in the early 20th century, it was not clear whether these spiral nebulae, as they were known, were simply part of the Milky Way or whether they were island universes unto themselves. Two great astronomy pioneers who had Wisconsin connections, Henrietta Swan Leavitt and Edwin Hubble, revolutionized our understanding of the universe. Henrietta Leavitt lived in Beloit, Wisconsin for a time, where her father was a congregational minister. She even taught art at Beloit College for 2 years. She was one of the Harvard computers, a team of women who worked at Harvard Observatory doing astronomical analyses. Leavitt observed variable stars called Cepheids in the small Magellanic Cloud. Cepheid variables brighten and fade in a periodic way, which corresponds to the oscillation of the size of the star. Leavitt discovered that the brightest stars, so the ones at the upper part of this diagram, had the longest periods. This is now known as the Leavitt law. Hubble did research at Yerkes Observatory on Lake Geneva in Williams Bay, Wisconsin. He observed that Cepheids in the outer parts of the biggest and brightest of the spiral nebulae, Andromeda, were much fainter than any stars in the Milky Way, despite their intrinsic brightness. He measured their periods and, using Leavitt's law, their brightnesses. He made the discovery that the Andromeda galaxy was 2.5 million light years away and not part of the Milky Way, which is only 100,000 light years across. He carried on this work by measuring the distances and recession velocities of galaxies, and he made the astounding discovery that the universe is expanding. I like this cartoon in the lower left corner. We have a stockbroker saying, if the universe is still expanding, get me some. If anyone would like to invest in the universe, please see me after this talk. So here's Hubble's original data, and his law is at the top. Velocity equals h naught d. It's known as Hubble's law, and h naught is known as Hubble's constant. It's the rate at which the universe is expanding. By measuring the distance between the Milky Way and another galaxy, such as this diagram is illustrating, and by measuring the speed that galaxy is moving away from the Milky Way, we can calculate how long the two have been separating using the relation that time equals distance over velocity. This is the same relation that you would use to calculate how long it will take your friend to drive home after visiting you, assuming he's traveling at a constant speed. If we replace v with Hubble's law, then we find that the distances drop out and we get the Hubble time, t equals 1 over h naught. This is the elapsed time since the Big Bang. We have managed to measure h naught, which means we know the age of the universe, and it's 13.8 billion years old. Thank you.
Right, thank you. Questions? Laura, do we have any questions online? Questions from the audience, please step up to the microphone. Oh, Hubble time is stretched here.
What's the evidence that the velocity is constant?
It's not. So in fact, there was an early deceleration of the universe and a more recent acceleration of the universe. And the two kind of compensate that the Hubble time is roughly right.
Will we ever be able to like travel at the speed of light?
No, we will not, because we have mass. Well, a photon can travel at the speed of light because it is light, but it is massless.
All right, next question. Oh, yeah, step right up.
So you mentioned how the universe is kind of accelerating away from each other. As the galaxies spread apart faster and faster, and as the light from those galaxies redshifts into basically unobservable states, will there be a time when we can't make this observation anymore?
That's correct. It's going to be a very depressing future. We were lucky we lived when we did.
We do have one online.
Yes.
Can you hear me? Is that okay?
Yes.
Yeah. How do we know that the two galaxies broke apart at the same speed at the time that the Big Bang happened?
So these galaxies did not exist at the time of the Big Bang. So they formed later in time. But we know that the universe, the expansion of the universe is space-time itself. It's not the objects themselves. So they're not physically, of their own volition moving away from one another. They're just being carried along with the expansion of space.
What's beyond the expanding edge of the universe?
Well, unfortunately, we are limited in how far we can see. So we have an observable universe that keeps growing over time, but we have no observations beyond that. So there's lots of theories out there, lots of nice speculation.
Well, we know that the Hubble telescope is going to be replaced soon. What will the new telescope tell us about the expanding universe?
I think so this is called the James Webb Space Telescope, and it's going to be launched later this year, December, I believe. So it's very exciting. And it's really going to be probing the earliest galaxies. So we're going to learn about how those earliest galaxies formed. There's a theory that galaxies form hierarchically, so they're smaller early on and they get bigger and bigger as time goes on. And we have observations that support that and theory that supports that. But we'll be learning more about those little bits, those very tiny pieces in the early stages of the universe, because you're observing at near infrared wavelengths, which is, because the light's getting stretched due to the expansion of the universe. We can only study these at longer wavelengths. It's hard to see them at optical wavelengths.
So given all these caveats, is your guess that the universe actually is likely to be older than 13.8 billion years?
No, this is the age of the observable universe.
Any other questions? You've got a minute and a half. Burning questions you have for an astronomer. Oh, go hop back up to the mic.
The new telescope, will we be able to detect, I can't say see because we're not going to see anything, but can we detect the older galaxies a lot better or not?
Well, so you have to be careful with the word older. So, I mean, light takes time to reach us. And so we will actually be seeing the galaxies when they're very young. And so, like I say, we'll be seeing them sort of before they've become these grand design spirals. I mean, they're just going to be these little irregular, tiny clumps of stars at these early stages. And so we will learn more about how the early galaxies first formed. But the present day galaxies are sort of the old galaxies because they've been around so long.
How many significant digits can you use to calculate the age of the galaxy?
The age of the galaxy?
Or I'm sorry, the age of the universe. I'm sorry. How many significant?
Well, I should remark that there are some uncertainties in the measurement of H naught, and that there's a popular tension right now that people are pondering. But I think this mostly probably 13.82 billion years or something like. I mean, I'm not sure about how accurate it is.
All right, thank you, Amy. All right, we will reset the clock, and Phil, the stage is yours.
Okay, I'm going to show you something amazing. You're watching a movie of a fertilized newt egg undergo its initial embryonic cleavages. One cell will now convert itself into a complete organism. You've seen the early stages of development where the cells are dividing. Now we're going to zoom in on the dorsal lip of the blastopore here. This is the process of gastrulation as cells flow into the embryo, turning this hollow ball of cells into the 3 tissue layers that make up the vertebrate body. What you can now see is the formation. That was wrong, sorry. I was going to show you the beautiful neurulation where the nervous system is formed, and then you'd see a beautiful tadpole hatching out. My time is not going to let me do that now.
Stop, it's so great. We have to.
Okay. Thanks, Laura. No, one more. Sorry. Okay, here we go. And I think okay, so we're going to watch these cleavage divisions, and we'll start me once we get to neurulation. Does that sound good? So again, we're going to watch this. So this movie, I should tell you, is available on YouTube. Jan van Eycken is the photographer that filmed this. He did just a beautiful job. It took him many months to capture this about 3-week-long process. So we're back at neurulation here. I'm sorry, gastrulation here. And now we're going to watch as the nervous system forms. So now you can start me up again. So basically what you'll see is this tissue flattening out. And this is the neural tube forming and then closing. This is giving rise to the whole central nervous system of this embryo. The brain is forming up here. The spinal cord is down here. And then you can see the guys, his muscles are now starting to twitch. And within 3 weeks, you form this beautiful little tadpole, which you'll get to see emerge from its egg capsule. Okay, so if I do anything, if you go home and watch this movie, I'll have done my job today, because this does more than any, like just watching this amazing thing is the best thing I can have you do. So in terms of how we know how animals develop, the first approach that scientists took was simple observation. Going back to Aristotle, opening up a chick egg and looking at the embryo inside, as microscopes got better, scientists were able to now view these processes more clearly. You're looking at a C-squared embryo here, and Conklin found these beautiful orange cells. This pointer is not working. There's a cytoplasm that is inherited, that's visible by pigment, and the cells that take up that pigment wind up forming the musculature in these larvae. Moving beyond observation, scientists then began to experimentally manipulate embryos. So the Ascidian embryo that we looked at before, if you ablate 2 of the 4 cells on the left of the embryo, you'll recover a half embryo that just represents the right side of the animal. In these famous transplant experiments of Spemann and Hilda Mangold, if you transplant the dorsal lip of that blastopore that we saw earlier from an unpigmented newt to a pigmented newt species, and you add it to the place that would normally form skin of the embryo, what Mangold saw was something remarkable: the formation of an entirely new embryo along the side. And when she looked at that tissue, what she could show was that cells that would normally make skin actually were now signaled to make nervous tissue. So these 2 different kinds of experiments suggested that embryonic fate could be determined by a cell's parents, or embryonic fate could be determined by a cell's neighbors. All right, so in terms of figuring out the mechanisms of how this all worked, that required molecular genetic analysis of the simple fruit fly. There are amazing fly mutants that have converted antennae into legs, for example. This is the Antennapedia mutant. The ultravithorax mutant converts this thoracic segment that normally has these little balancer organs into a whole second thoracic segment. So the fly now has 4 wings instead of 2. These are called homeotic mutants that transform one segment into another. Additional screens identified all kinds of mutations that affected the patterning of the fly larva, identifying mutants that change the kinds of segments, their numbers or polarity. And now when scientists went to look at what these genes were that were behind these amazing mutations, they saw that these genes, both Antennapedia and ultravithorax, encoded transcription factors that could turn on the expression of other genes. And it turned out when you looked at where they were on the chromosome, they were actually organized in a way that reflected their expression along the head to tail axis of the embryo of the animal. And to everyone's surprise, these genes were not unique to flies, but are shared throughout animals. And in mammals, we have the same group of genes, except we have many copies of them, also arranged linearly along the chromosome in the order in which they're expressed in the animal. And so the genes that are defining regional identity are conserved between flies and mammals. So basically, all of this work has told us that different animals use the same molecular toolkit to drive embryonic development. And we've learned all of this by studying simpler model organisms like flies, worms, frogs, and mice, which would be another talk. Woo!
Best movie of the Science Festival by far. Questions? Step right up.
This may seem a dumb or ignorant question, but if you do some experiment where you end up with a fly with 2 sets of wings, does it pass that on to its offspring?
So those are absolutely genetic. Those are mutations that allowed people to really define that. So these are genetic mutations. Of course, there are other kinds of perturbations where you could imagine messing it up where it wasn't genetic, but that's really what allowed us to actually make these incredible observations, was that people had spent over a century using Drosophila to understand first just the basis of how genes were inherited. That was the first Nobel Prize for Drosophila. But later work then used those tools of genetics to actually use mutations to understand the process. And that's how that gave us the inroads to actually understand this incredibly complicated process. Because prior to having mutations that disrupted it, there wasn't really a way to even think about how you could study this. So people really thought that embryonic development was too complicated. We would never get a handle on it. But by taking mutations that could disrupt very specific processes, that then allowed us to have this molecular insight. And so one of the amazing things that has emerged, so I gave one example of this whole group of these homeotic genes. Several of those genes that affect the patterning of the fly larva, and they're just identical, the little hairs on their ventral surface, turn out to be incredibly important signaling molecules that play roles in human cancers and in human development, in the setting up of our proper anterior posterior patterning of our limbs. Other genes have been found that control heart development in both flies and us. And actually even the fly eye and our eyes use the same kind of master regulatory gene to activate gene expression. So in fact, one of the genes, major regulators of eye development that was identified in Drosophila turns out to be mutated in humans that have a disease called aniridia. And the human gene can actually turn on the fly genes and make a fly eye in the fly. So when politicians talk about the uselessness of fruit fly research, I hope you understand that that's really wrong, and that actually we've learned a tremendous amount about biology by studying these simple creatures that have really allowed us to gain insights into things that would have been far too complex, like ourselves.
Another question. Yeah, hop on up.
Okay.
Oh, there we go.
So with enough mutations, can the fly become a different organism?
So that's a great question. So I think, I mean, that you're getting at a really profound question, which is these kinds of mutations that affect patterning. You know, I've told you that they're shared between fruit flies and us. So how are we different? Well, we're different in the way we deploy all these different kinds of what we call toolkit genes. So absolutely, by changing segment identity, right there. So the ancestral insects had 4 wings, right? And so the hind wing was basically controlled by certain genes. And now when this gene began to be expressed in the 3rd thoracic segment in Drosophila, that basically shut down that hind wing difference. And so those kinds of things that you see in all the various different kinds of insects, for example, that have different hind wings than forewings. So how those things are deployed will be critical for generating new species. Great question.
Laura, any questions online? Anybody else? Oh yeah. That's what I like to see.
So is it known what maintains the anterior to posterior ordering of the homeobox genes, why they're so conserved in that order functionally?
That's a phenomenal question. I think people are. Oh, sorry. Sorry, he's a ringer. I don't know what he's doing here. So the question is, so I told you that those genes are basically oriented on the chromosome in the order they're expressed in the animal. So that the genes on the left are expressed in the anterior and the genes on the right are expressed posteriorly. People are actively investigating that. I actually am not, I don't know that we have insight into that yet. So it's a great question. It's something that is super interesting.
Oh, you're gonna be fast.
How do fruit flies all of a sudden end up on your bananas? I mean, where are they coming?
They're just, yeah, that's a great question. They really like being around humans, and they're really good at finding bananas. Yeah.
Expressed as a gene, finding bananas?
What's that?
Is that expressed as a gene, finding bananas?
Sure, it is. They're absolutely.
All right, thank you. All right, we've reset the clock and turned it over to Hilary.
Hello. A couple things to do here. All righty. My name's Hilary. I'm a limnologist. Show of hands. You know what limnologists do? Madison, I love you. This is I think per capita, the only city who knows what limnology is. But it is the study of inland waters, and I think a lot about lakes as a chronicle of our changing planet. And in limnology, we often refer to lakes as sentinels, this idea that they're sitting there watching what's happening. Lakes are phenomenal to study because they integrate everything around them. They integrate what's happening on the landscape, what's washing in from the watershed. They're interacting with the atmosphere. And so they're really changing, not just from climate change, they're changing for a whole number of reasons. And while oceanographers get all the credit because there's takes up like 75% of the planet, there's hundreds of millions of lakes on Earth, and they're just sitting there absorbing what's happening around them. And so we can understand climate change from the lens of these 100 million lakes. And so going to focus on two examples today, and we can start right here, Lake Mendota, near and dear to our hearts. And every year, Lake Mendota freezes. And this is something that we've come to appreciate. The lake freezes, we can go out and recreate on it. By March, we're getting pretty tired of lake ice and we want it to go away. And we have one of the longest records of lake ice dates that exist on the planet. People in Madison started measuring this in the 1800s. And what we can see in this top graph is just sort of there's a dashed line for the ice duration through time. And so back in the late 1800s, it was common for the lake to be frozen for 4 months of the year, which maybe we wouldn't appreciate nowadays. Then there's a lot of years where we see 3 to 4 months a year. And as we get closer to present day, we're starting to see more like 2 to 3 months a year and sometimes less than 2 months a year of lake ice. And so we're seeing this change in lake ice, which is indicative of something changing in the climate. And we can see another sort of representation of this change in lake ice. In the 1800s, we had a much longer duration. The lake was definitely frozen to December. Sometimes there was ice till May. And now that window is getting shorter. And there might be some day where there isn't lake ice on Mendota. And as I said, there's hundreds of millions of lakes on Earth. Many lakes are in climates that freeze. And so lake ice is a really good indicator of change. And so if we look not just at Lake Mendota, but at lakes all over the world, we can see that this duration of lake ice is changing. And this is one of the clearest indicators we have globally that this is happening worldwide. And spoiler, there's no lakes where lake ice is increasing in duration. And so these dates of freeze are getting later in the year, and the dates of ice off are getting earlier. And not only is this an indicator of climate change, but it's also having ramifications for a lot of other ecosystem processes in lakes. So that's winter, one season. In Wisconsin, it happens to be the season that's warming the fastest. But we can also think about summer. And limnologists have deployed buoys like this all over the world. This is David Buoy on Lake Mendota. If you've ever been out, you see him floating there. Basically, he goes in once that ice comes off, stays in till about Thanksgiving. And we have a lot of sensors that are on these buoys down to the lake water column. We can monitor change at frequencies about every minute. And we also can see what's going on in the atmosphere with some of these sensors on top. And if we look at these records from all over the world, we see that globally lakes are getting warmer. And so this top graph is surface water trends globally. We can measure these both in situ from sensors. We can also measure them from satellites, just the sort of skin temperature on the top. And we can see that there's a lot of red dots. And so lakes are getting warmer. Here's some long-term examples from some lakes in Austria and the UK. You can also see there's some blue dots. Feel free to ask me about those if you want. There's some lakes that are getting colder for different reasons. And this is another indicator of change. And so lakes worldwide are telling us that the climate is changing. Air temperatures are getting warmer. Precipitation is changing, which is also having other interesting effects. And the ramifications of this are huge. Higher temperatures mean less water, there's more evaporation, so we're losing surface water in some of our reservoirs. The US West is a perfect example of that. And then closer to home, we're thinking more about algal blooms. So lakes like Lake Superior, traditionally pristine water, we're seeing more blooms and trying to understand how that was related to climate change as well. So 10 seconds left, and I'm happy to answer questions on lakes and climate change.
Questions? Yeah, there we go. Come on up. You can hop in.
Tell us about the blue dots.
Yes, so some lakes are getting colder. And one region in particular that people talk a lot about is on the Tibetan Plateau, where actually there's been such a recession in glaciers that there's a lot of very cold glacial meltwater. And so because glaciers are actually melting at higher rates than they had previously, that cold runoff water is actually cooling the lakes. And so those lakes have gotten colder just because the glaciers are disappearing. So interesting feedback of climate change that you weren't suspecting. And then in other parts of the world, it's actually due to clarity. So sometimes with reforestation, the lake water is actually getting clearer, which causes heat to penetrate deeper, and it mixes in that upper layer and cools down.
What's a lake?
Oh, good question. One time I interviewed and I was talking to a cloud physicist and he was like, "What's a lake?" And I was like, "What's a cloud?" I mean, I don't know. Oh, what is a lake? It is a depression in the earth that usually has water in it. And it comes in many forms. Some people try and differentiate ponds. There's no real definition. You can consider ponds lakes. I could think of lakes as not freezing solid in the winter, but that only works when you're in Wisconsin. So that definition does change. And how do you count all the lakes on Earth if you're counting little ponds? That number just gets episode. It's like trying to figure out how old the universe is. It keeps going. It just keeps going.
In addition to the lake ice on, ice off, and Lake Mendota and Monona, are there other changes in, say, the quality of the ice, the strength, the thickness?
Yeah,
the shape?
My answer is probably we're really bad at measuring that because it's harder to measure. The ice on and off dates go back so long because it's just we can observe that, and things that we can observe with satellites is their ice. And so actually getting out and testing the quality of the ice is more challenging, especially in those shoulder seasons where the getting physically on to lake ice is really dangerous. And so that's really prevented a lot of research from happening in the winter in general. So we've been as a field very summer biased. I mean, it's nicer in the summer to get out on lakes, but that's a really a question that hasn't really been approached yet because we don't have all that much data on ice quality. Yeah.
Laura.
We have an online question. Does increased runoff with nutrients affect how long the ice lasts?
Probably not is the short answer, because it would be going underneath the ice and not really affecting the ice quality at all. If your lake gets salty enough, that will change your ice quality. And so there are places that are so impacted by human salt input, especially from road salt, that it depresses the freezing point of ice and can create differences in ice quality more than nutrients. But nutrients have their own bag of problems. So
we've talked a lot about how climate affects the lake. Can you talk about how the lake affects climate, like the locale around it?
Yes. Well, so one thing we think a lot about is carbon dioxide, which is a potent greenhouse gas. And overall, lakes actually emit carbon dioxide, so they are contributing to greenhouse gases in the atmosphere. Coincidentally, when lakes have increased nutrients and we see these big algal blooms, algae are like trees, they are photosynthesizing and taking in carbon. And so one of the minor benefits of poor lake water quality is you're actually sucking in some CO2 from the atmosphere. So, you win some, you lose some.
What are some of the biggest impacts you see on the living ecosystem of the lake with the shorter ice period?
So the thing that people think about a lot is just habitat. And so thinking about fish habitat. So fish are adapted to certain temperatures. So some of our favorite fish species are cold water fish like walleye and cisco. And so as lakes warm, you're just losing that habitat and you're stressing fish and pushing them into narrower sort of bands that they can survive in. And so as lakes warm, we're just going to lose those cold water fish species. And so there's been lots of work, especially in rivers, to try and protect temperature just by like trees. Shade helps. In a lake, obviously, that's just harder. Yeah.
What's your favorite lake?
My favorite lake is Lake of the Woods. Any fans? It's a pretty good lake. So it's a binary. It spans the border. Yeah.
3 seconds. When is the lake going to freeze this year?
January 12th.
Okay, you heard it here first. All right, thank you, Hilary. All right, next up, Keith. We'll reset the clock and we'll get you started.
Okay, so of course, figuring out who done it is one of the central enterprises of the criminal legal system. And like science, it depends on evidence. But one would think that we would have a fair bit of skepticism about our ability to get it right, given that unlike science, this is an entirely human enterprise with not a lot of good measurement tools or ability for testability and whatnot. But where I want to start my story is to take us back about 30 years or so and think about what our view of the criminal justice system was back then. And by and large, that view was this pretty self-satisfied assumption that we got it right pretty much all the time. And we saw this in expressions in Supreme Court opinions and throughout our society, that we felt like we bent over backwards to protect criminal defendants. So sure, as a human system, we would make mistakes every now and then, but those errors had to be so rare, so freakishly rare as to be of little concern. That all began to change with this. The development of forensic DNA profiling, which came along in the late 1980s, and for the first time ever started to prove that our system was getting it wrong, and getting it wrong in important high stakes cases and on a far greater scale than we ever saw. The data today, which largely comes from the Innocence Project, tells us that since 1989, when we had the first DNA exoneration, we now know of at least 375 cases in which a stone cold innocent person was wrongly convicted of a crime. And these are all really serious crimes. 99% of them are rapes and murders. 21 of these people have been sentenced to die for these crimes that they didn't commit. As you can see, they've served an average of 14 years in prison before exoneration. So the consequences are really, really harsh. But this is just the tip of the iceberg, because this is only those cases where the fortuities all came together to exonerate somebody on the basis of DNA. And DNA, as wonderful as it is, doesn't exist in most criminal cases. So a group of scholars have tried to identify a broader pool of exonerations, and they've created this National Registry of Exonerations. And since 1989, they have counted closer to 3,000 exonerations on the basis of any kind of evidence, not just DNA. Now, let's figure out how many people do we think there still might be in prison. Now, this is a really hard thing to measure, but we know that there's about 2.3 million people behind bars in this country. The best empirical estimates that we have to date are that perhaps 1 to 5% of those are wrongful convictions, that those people are innocent. Well, if you do the simple math, that means we've got anywhere from 23,000 to 115,000 actually innocent people languishing behind bars. So one of the great things about these cases, though, is that they not only tell us about the scope of the problem, but they allow us to study the problem and identify what is it that leads to wrongful convictions. And we know that one of the leading contributors, depending on what pool of cases you look at, is simple eyewitness error. Surprise, surprise, people lie, right? So 61% of the cases in the National Registry involve various kinds of perjury. This one is a surprise because it's so counterintuitive, but about nearly a quarter of the DNA exonerations involve people who falsely confess to crimes we know they didn't commit, serious crimes. And then there's flawed forensic science evidence. Isn't that ironic? It was forensic science in the form of DNA that came along and exposed the errors, and only to turn around and realize that forensic science was part of the problem also. And what are the problems here? Well, if you look at, for example, as researchers have done, at the testimony in wrongful conviction cases, cases where a person was proven innocent, and they look at the transcripts of the actual forensic science testimony, they see that in 61% of those cases, the forensic science testimony was scientifically invalid. This was all studied extensively in 2009 by the National Academy of Sciences that issued this report, and their conclusion was earth-shattering. With the exception of nuclear DNA analysis, no forensic method has been rigorously shown to have the capacity to consistently and with a high degree of certainty demonstrate a connection between evidence and a specific individual or source. And this includes all of the old standbys, the standards, fingerprints, shoe prints, bite marks, you name it, firearms, all of these, it turns out, have no scientific foundation, and the error rates are entirely unknown. They are largely subjective enterprises. It ain't CSI. And because of that, very little has been done, and unfortunately, to remedy this problem. Dr. Handelsman here has been, when she was in the White House, was starting to work on those kind of problems. Unfortunately, there was a change of administrations that put an end to that kind of inquiry. And so now my colleagues and I, including Joe, have created this center to work on that problem.
Fantastic. Questions?
Any guess as to why there's so much resistance to making it right when we discovered we've made it wrong, we've done it wrong?
There's a lot of factors that go to work. A lot of it stems from the fact that we have an adversarial system. And so people have entrenched interests. And in particular, when I'm talking about prosecutors. One of the great resistances to, for example, a report that Dr. Handelsman's office produced, which is something known as the report of the President's Council of Advisors on Science and Technology, was reports about ways to improve forensic sciences. And President Obama's own Attorney General, the chief prosecutor in the country, disavowed the report, refused to follow it because of a concern that actually employing scientific procedures would make it harder for them to get convictions. Correct me if I'm wrong.
Other questions? Yeah, come on up.
So with the method that we've been using and they've been proven inaccurate, then how would you convict someone?
Well, the idea is that all of these methods are, what they've shown us is that they're fallible. But you're right, we have to depend on evidence. We have to depend on eyewitnesses. We need forensic science. Confessions can be useful. The trick isn't to get rid of them all, but rather to find ways to make them more reliable and accurate. So a lot of study has been done, for example, on eyewitness identification error, and it turns out that the way that the police conduct identification procedures can greatly influence the accuracy of the ensuing identification. So if police are attentive to making sure that they put together lineups that are not suggestive in their nature, that are conducted double blind, like any scientific test would be done, they provide non-suggestive instructions to eyewitnesses, all of those kinds of things can dramatically reduce the rate of error. And you look across the board at all of these flawed kinds of evidence that we rely upon, and there are ways to make them all better. We will never achieve perfection. We will never have an infallible system, but we can do a lot better than we have been doing.
Yes, come on up.
A while ago, I served in the jury for a criminal trial, and the defense tried to submit evidence that was dismissed as hearsay, and so we were instructed to disregard it, but you can't just forget it. So I'm wondering, like, what is being done or what can be done to kind of generally educate the public about like what is good evidence?
What can be done? Well, you're right. You can't unring the bell. And guess what? Judges and lawyers know that too, but because it would so muck up the system if they had to start over again every time there was an error made and somebody was instructed to disregard the error, we engage in this legal fiction that the jurors will follow that instruction. They can't. But the bottom line is, there are several things that can be done. One is education, like what we're doing here tonight, just to make sure that our jurors as decision makers come to the enterprise better informed. Other things that we can do, though, is to present expert testimony from people who've studied these problems to help jurors understand what makes an eyewitness identification more unreliable or less so, or what leads to false confessions. Do we have those things going on here? And even jury instructions from the judge to educate jurors about those. So there are things that can be done sort of on a societal basis. There are also things that can be done in the courtroom to help jurors be better decision makers.
How reliable are eyewitnesses?
Eyewitnesses are not very reliable. It turns out that human beings just are really bad at recognizing the faces of strangers. And there's a lot of data on that. You know, it's a little bit difficult to test because in the field, because in order to know if an eyewitness has made an error, you have to have access to ground truth. And in most actual casework, you don't know. But one way we can tell that witnesses are making mistakes is to look at those instances in which the witnesses in real cases pick out fillers. That is, people who are not suspects at all, but are just plugged into the lineup to round it out to make it a fair test. And usually somewhere in around 20 to 25% of the cases, eyewitnesses pick out known innocents. They pick out fillers. The rate of error, though, depends in a tremendous degree on the nature of the procedures that are employed. So the less suggestive the procedure, the more blinded the procedure is, the much more likely we are to get reliable results. But there is no one number we can give you because there's so many variables at play in each identification procedure.
Anybody else? All right, thank you.
Thank you.
All right, we will reset and have our last speaker of the evening.
Much good luck with your procedure. Thank you.
Yeah, take it away.
You just advance the slide.
Oh, I click it, sorry.
Yeah, yeah.
Glad you said something. Okay. So I'm going to talk tonight about where fungi are. And a really short answer to this question is everywhere. And I'd like to try to make it a little bit more nuanced by asking the question, which fungi are in which places and how can we know? And again, a really short answer to that question is we're absolutely at the start of answering that question, and we don't really have a good handle, not in any way, shape, or form do we have a good handle on which species of fungi grow where. But I'll try to unpack why we don't and what's happening now that's really exciting. So this is high in the Colombian Andes, which is one of the places that I work. But now I kind of want to restart the question by focusing on this adorable chickadee. And birds are something that we know a lot about. We know so much about birds that even though this is just a chickadee that we're all familiar with, there are like entire books written about just these few species. And you can hear the irateness in my voice, and I'll try to unpack that a little bit as we go on. So the reason that we know so much about adorable chickadees is because they're so cute and because we can see them. And as humans, we have a lot of biases. And one of our biases is that we're visual creatures. So things that are beautiful and have interesting behaviors that we can watch, we pay attention to them. I'm a closet bird watcher, but it sometimes drives me crazy that we not only write books about things like chickadees, but we have like granular data from heaps of people who go out and want to tell you about the chickadees that they saw in their backyard during the Christmas bird count. And so not only can I tell you that we know heaps about chickadee ecology, but I can even tell you that like in Texas, there are very few chickadees. People have counted the individual birds. People have counted the birds in Wisconsin, and it's not showing up quite there, but you can say that there are more chickadees, more individual bodies of chickadees in the north of Wisconsin versus the south. And I can do this for almost any bird that you care to imagine. Bald eagles, sandhill cranes, whooping cranes. I can tell you where they are. I can tell you what number are there. I can tell you so much about them. I could write a book about a crane or a bald eagle. In fact, I'm sure there are books about bald eagles and cranes. So when we turn to fungi, here's the state of the art. Old 1800 style, beautiful, lovely illustrations of mushrooms. Here's one that first appeared in New Zealand in the 1970s. Finally, the mycologists in New Zealand decided that it was an introduction from Europe. In Europe, they had this fungus, but they thought it was an introduction from New Zealand. It's still the subject of an intense debate with the Europeans being accused of being colonialists and the people in New Zealand. Anyway, it's kind of a mess. But so for some things, we have data like this, a description from a range where the species has been introduced. And we really don't know where it's from. We don't know the native range. But most fungi don't have those beautiful mushroom structures. They look like this, little fuzzy dots on a petri dish when you isolate them out of water. But the tool that's happening now that's really exciting is, and you've already heard about it multiple times tonight, DNA. So this is a bog or a lake, I'll let Hilary decide, in Massachusetts, and you're seeing the outline of it there. And these are species that grow inside the pitchers of carnivorous pitcher plants. So these are like little test tubes out on the landscape, and we can go along and we can sample the fluids out of those pitchers, and we can sequence DNA. And by sequencing the DNA of this Candida and Rhodotorula and Pseudozyma, we can say at 4 days into the season, this is where the Candida is. 1 week, this is the pattern. 1 month and 2 months. So now just for these 3 species represented as fuzzy dots, which is more or less all the visual you're ever going to get on these species, I can give you really precise information about their dispersal patterns across this lake/bog in Massachusetts in 1 season in summer. So with DNA technologies that enable you to identify the species that you cannot see, you can start to unpack patterns of biodiversity. However, the problem with this approach is scale. So what you're seeing on your left is this massive, what's called a phylogeny. And the way to think about it is it's a graph of all of the species that are found inside of a certain collection of pitcher plants, not even very many pitcher plants. And you can see it's a complex graphic with a lot of lines. How do we scale the diversity that's inside this small habitat, the pitcher plant, to the diversity across the globe?
All right, thank you, Ann. Questions? We have one online, Laura? Questions?
But I have
Can't
one.
think
Can
of
I
it.
cheat and?
You can cheat, yes.
Would it be like Professor Hawks talked about for humans? Is it possible that all fungi have a common ancestor?
Oh, all fungi. Yes, it's definitely true that all fungi have a common ancestor. There's interesting discussions about what that common ancestor looked like. But we, unlike with the world of animals, I can't tell you how many species of fungi there are on planet Earth. So estimates range from 1 million to 10 million, so over an order of magnitude. And those estimates are based on various kinds of extrapolations that are all kind of inherently flawed and problematic, but it's sort of where we are. So and most of that diversity is undescribed. So we have names for something like 5% of the species of fungi that we think exist on our planet.
I have a lot of mushrooms in my yard this year, unlike other years. So, and they seem to grow like that overnight.
Yeah.
What is it about this year that made it a good year for them, and how fast do they really grow?
That's interesting that it was a good year for you. For a lot of people, it was really a terrible year, actually, because there was not a lot of rain this year. So the seasonality of when fungi appear has to do with many things, the things you would think of like rain, also where they get their carbon from. So a fungus that's tied to a plant, often the mushrooms form when the plant drops its leaves. And so as the plant is shutting down and taking its carbon back, that's when the fungi appear. But there are, of course, but most of the fungi that exist on Earth don't make those beautiful mushrooms. So that's a really rare subset. And I'm not even sure I answered your question. But how they appear overnight is also a really interesting question. I think that was part of your question. And the best idea is that they're sort of pre-formed. The cells have divided, and there's, if you want, a miniature mushroom underground, and like one of those toys that you buy at a kid's store where you add water and the dinosaur pops out as a sponge, that it's something like that, that the little mushroom absorbs water in the right environments and then it pops out. Because they appear practically overnight.
How about, yeah.
People were getting tired of looking at me at the microphone. I'm more of a botanist, and I've taken mycology before. And when I'm looking at the ground or digging into the root systems, one thing, and then backing up and then thinking about the larger picture, the species interactions, that is, the food web interactions, if you were to be able to total all of that, yeah, if there's 1 million or 2 million or 3 million species, so what? But they're so integral to everything else in the ecosystem. And that amazes me. How much research is going on right now in individual areas that try to put that complexity together in one package?
Yeah, it's a super question. So for my entire career, I've been practically the most obscure person I know because I work with fungi, and most people had never heard of fungi. And you may have to explain what a lake is, but I kind of had to be like the spokesperson for a kingdom, which is nobody ever asks someone who works with animals, how does an animal disperse? No one ever asked that question. They ask, how do birds do it? How do fish do it? How do insects do it? But when you work with fungi, people are constantly asking me, how does a fungus move? Or why does a fungus do this? Or da 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 da. And it just showed, so there's a huge gap in knowledge. The good news is that with new tools like DNA research, our toolkit has exploded. It's not just DNA. There's lots of different new kinds of tools that we can take advantage of. Computers, supercomputing, ways to analyze massive amounts of data that we could never analyze before. So the field is exploding. It's a huge moment of growth. I have more people applying to my lab than I've ever had apply to my lab before in my entire career. So I think it's I would say the news is encouraging. First of all, I don't have to explain fungi the way that I used to. So I think somehow it's a cultural moment where people are learning about them, even the whole science festival being devoted to that. People are learning about them. People are curious. People want to know more. People have new tools. So I think it's kind of a great time to be a mycologist. Yeah.
Any other questions? Laura, anything online? Anybody? Anybody? You're gonna get off easy, Ann. Oh, wait, we've got one coming.
How many fungi are on the chickadee?
A lot. There's a woman who's joining, Michelle Giussino is joining the Forest Service in a few months, and she has this incredible body of work that looks at woodpeckers. Did you know that woodpeckers carry fungi on their beaks? And when they're pecking their nesting cavities, they're actually enabling the decay of their nesting cavity by carrying fungi around on their beaks. So it's a really, yeah, that feels like a wonderful box to unpack. Almost anything you look at in the world, if you ask, where are the fungi? As I said, they're there. Yeah.
Thank you, Ann. Let's have a round of applause for all of our speakers tonight. So you've been a really, really great audience. Thank you for coming out. There's more fun this weekend. So stick around for more fun this weekend. We've got Science on the Square. Laura's going to advance a slide for me. I'm getting editing. We've got Science on the Square tomorrow starting at 5 o'clock all along State Street. Select locations around the square. You may stumble into a scientist at various bars around the square. You might see me if you look carefully enough. Lucille, 6 o'clock, just noting. We also have back here on Saturday a lot of room with the Wisconsin Book Festival events. We've got 2 panels. Where are we? Where are panels?
There's a panel.
There's actually go back one slide to the book festival. I get all the time in the world. The clock stopped running for me. Great at Discovery Building on over the weekend. A bunch of great book talks by a lot of our faculty at UW-Madison talking about some really great things. So I encourage you to come back to that. And we've got, of course, Magic Mushrooms, an event at 12:30 hosted by Steve Paulson from Wisconsin Public Radio. He is going to moderate a conversation on magic mushrooms, AKA hallucinogenic. I got to get that right. Hallucinogenic fungi and the New Research Institute here at UW-Madison. So I encourage you to come back for that. And at 1:45, we'll hear all about fungi in Wisconsin, the good, the bad, and the ugly. It's all good though, isn't it, Ann? No. Okay, all right. Good. And on Saturday night, you can still sign up for a free double feature movie night at the Madison Mallard Stadium featuring fantastic fungi, followed by Alice in Wonderland. So come on out Saturday night for movie night. Have a great evening, everybody. Thanks for coming out. We'll see you over the course of the weekend.